हाँ बस आपको रिकॉर्डिंग अच्छा ठीक है आप फिजिक्स के हो यस हेलो हेलो जी अच्छा सेटिंग जो है एक मिनट ये कौन से वाला है स्पीकर यही है ये जो है सेम सिस्टम ये वाला हाँ हेलो हेलो अब हाथ जो है अभी अनम्यूट है हाँ हेलो 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 वो वाली स्पीकर भी हमें चाहिए ना जो सिस्टम वाले हैं जो अब चल रहे होंगे यस अब बोला हेलो हेलो पता नहीं हाँ हाथ कैन यू हेयर अस प्लीज अभी रिकॉर्डिंग में स्टॉप कर देता हूँ फिलहाल के लिए
measure electrical recording from the brain. So just to give you a little bit of background of what this talk is going to be about, I would like to show you a small 1-minute YouTube video from Neuralink. So Neuralink is a company which was made by Elon Musk in 2017, and their goal is to commercialize brain implants and make brain implants accessible to like millions of people. So let's
for brain implant science. Academics like me have conducted clinical trials.
Can you guys hear the audio, right?
yes we can sure
Activity. So I will just now give a quick overview of the brain imaging systems. I actually work a lot on the imaging systems in the lab, and this is my primary work area of research that I work in the lab. So the first one I talked about was a hyperspectral reflectance spectroscopy. Here is the schematic diagram, and I'm just highlighting the optical path of this imaging technique. Here we have 3 wavelengths or 3 colors being reflected off from the surface of the brain with a glass window installed on the brain. And then there's an objective capturing the light with some dichroic mirrors to send the light to the electron multiplying CCD camera. We use these 3 wavelengths. This is very similar to the so what we're trying to do over here is we're trying to measure the concentration of hemoglobin and deoxygenated and oxygenated hemoglobin. This is very similar to what like we undergo when we go to the hospital, and then they put a clip on our finger to measure our SpO2 levels, or like if any of you have experienced with like these new Samsung or Apple watches, which can measure the SpO2 levels using your skin. So if you like flip the watch, you would see these are the same wavelengths that are actually using the green and the red light they're shining. So we know the spectrum of the oxygenated and the deoxygenated hemoglobin in the blood, and they're different at different wavelengths. Using this information, we can use a very simple absorption law, the Beer-Lambert law, which says over here that the intensity is exponentially being attenuated by the absorption coefficients. And if we like skipping a couple of steps over here of linear algebra, we actually get to this least squares matrix problem, where all of these coefficients in this matrix are known because they are in the literature. These are the molar extinction coefficients of these chemicals. the change in the absorption can be found experimentally from these images that I'm showing. The unknowns would be the change in concentration. So this is what we would be finding as a function of time and space. So at each pixel and at each time point, we will be finding the change in the hemoglobin, or deoxygenated and oxygenated. Why is this important, or what happens, like what actually goes on inside the brain? If I were to stimulate the brain, like for example, if I were to show some color to your eyes, right, or a visual, like a screen on a screen or something, then only the visual cortex of your brain would be activated, and you would see this localized activation. This is exactly what you're seeing over here. So as a function of time, you see nothing over here initially, but at t is equal to 0, I show some stimulation, like a visual stimulation to the brain, and then you see the brain would light up in a very localized region, and then it would go back to the baseline normal levels. So this is how the brain responds, and we have, so this field of view would be like it's around 2 millimeters to 2 to 3 millimeters squared.
Okay, Haad, these are just reflectances from the brain, which are spectrally filtered, or
Yeah,
there is also some fluorescence as well in there.
No, they're just pure change in the reflectance because of the change in the hemoglobin's concentration. So as the concentration changes, we would see a very, very small change. Actually, the percentage change you can see is only 3% or 4%. So that's the amount of reflectance that would change at different wavelengths. It would be different. For example, notice that at the 632 nanometer, you see always a positive change in reflectance, but at the other wavelengths, you actually see a negative change in reflectance.
Okay.
From these change in reflectances, using these least square fitting, we can actually find the concentration of the hemoglobin, deoxygenated hemoglobin. So this is one imaging technique that we use for seeing how the brain responds to different stimulations. So this is one measure of what the brain activity. Another common technique that we also implemented in the same system, so you can see this is the same optical system, which is being concurrently or simultaneously measured alongside the hyperspectral reflectance spectroscopy, is the speckle contrast imaging. So the speckle contrast image actually is just the this is just showing the raw reflectance, and it's being captured on the camera. There is nothing special over here about this, but notice that you can see the vessel over here slightly, the shadow of the vessel, and here you can see the speckle or the random fluctuations of the speckle pattern. So the way this image is computed on the right, the speckle contrast image, is that I think it did not power. Okay, so yeah, if you look at the blue trace, which is shown over here, and then the orange trace, which is shown over here, notice that in the orange trace, since we drew it on the vessel, so there is a unidirectional flow over here on the vessel, right? And therefore the intuitively speaking, we can understand that the fluctuation would be less. The fluctuation of the speckle would be much less compared to the blue spot, because the blue spot is where there is no flow or no velocity, no blood which is flowing in direction. So here, since there is a unidirectional flow, we would see the fluctuation is higher. And the way this image is computed is the standard deviation of the fluctuation divided by the mean. So when you do this, there would be much higher contrast over here. That's why it's white. And the contrast over here on the vessel would be much lower. That's how we compute from a raw speckle image, the speckle contrast image. It's very similar to basically the Doppler flowmetry imaging technique. You're only we're only measuring the places where the velocity of the blood flow is higher or lower. This image on the bottom left is exactly the same brain that I showed earlier for the reflectance spectroscopy here, and as a function of time. So here we show t is equal to 0 to t is equal to 8 or 9 seconds, and then we expose the brain to some stimulation. Could be visual stimulation, or could be like hand stimulation, or any kind of forelimb or any kind of stimulation in the brain. You see these white parts are actually our electrodes that are inserted in the brain. So this is why the image is being blocked over here. All of these white things and the black thing are electrodes. And here on the green is just showing the activation. So we can see that again, the activation rises and peaks at a certain time and then goes back to baseline levels.
अच्छा ये हाथ if you like जो आप analysis करते हो speckle analysis, so how many spectra you need to let's say construct the image, speckle contrast image?
So this is a single wavelength 785 nanometer single wavelength, very narrow band laser. And we're just measuring the interference pattern on the, because the brain, from when the light is reflected, it would create a random interference pattern on the camera CCD, and this is at the 785 nanometer wavelength, right?
So like the speckles are as a result of random diffraction on the image plane, let's say.
Yes, and then the way we construct the image on the right is purely based on fluctuations. Just wherever the fluctuations are high, it shows a white area. Where the fluctuations are low, because there is a very unidirectional flow, we would black contrast.
And then like how much, like how many how many spectra it takes to go from raw to speckle image, or just you can do it in from one image as well?
Yes, it's just one image. It's 1-to-1 mapping.
Okay, so then the question would be, what's the time resolution you can get with this method? Because you have shown spectra at t equal to 0 second, 1.7 second, 3 seconds. So what kind of resolution in terms of temporal resolution you can get with this?
So this naturally, since like the technique that I described is just the speckle pattern on the camera. So it is limited only by the camera frame rate. The camera frame rate we run usually at 70-80 FPS, but there are higher speed cameras as well, and you can go beyond that as well.
Okay, yeah, and then you post process it for
then we post
speckle.
And
Okay.
here, the way we actually are showing this transition is we're actually capturing images at 70 FPS. So there are like thousands of images being captured, and I'm just showing 4 of them over here.
4 of them. Yes. Okay. I got the point. Yes. Okay. Thanks.
Okay, and then talking about the 2-photon imaging, 2-photon fluorescence imaging. So normally the excitation wavelength for to probe the neural activity is actually around the blue region, 400 to 450 nanometers. But since 450 nanometer wavelength is actually not very penetrative and highly scattering in the tissue medium and the brain tissue medium, therefore we cannot, it's very hard to use this 1-photon single photon transition technique to image deeper inside the tissue. So if you want to go deeper inside the tissue, you want to actually image like 500 microns or 1 millimeter below the surface of the brain, you would want to use an IR or an NIR wavelength, and then use the 2-photon excitation. So the 2-photon excitation is a standard concept in AMO physics as well. Here, there is a ground state, and you want to get to the excited state. So you would, instead of just a single wavelength, like let's say the 460, you would break it up into 2 equal or unequal wavelengths. at very high power. So the transition probability, if you compute the transition probability of this, it's extremely low, and it scales with the electric field to the 4th power, or intensity squared. So therefore, we actually use a very high focus magnification, high numerical aperture length, and very high laser power. So the high laser power is achieved using the ultra fast, ultra short pulse like laser, so that we have a large energy concentration in a very small time point. So this is basically the diagram. It's a 2-photon is a non-linear optical technique, and this diagram is just showing there are other non-linear optical techniques as well, which we're not using, but we're only using the 2-photon just to describe over here. So since we are using the 2-photon, we use the NIR, the near infrared laser source can therefore penetrate deeper, and you can actually, I would like to show some of the results, what I mean. So previously, all of these images that I showed you are surface, just on the surface of the brain. We can't see anything deeper, but this is something that the 2-photon can achieve. So you're seeing over here the vasculature, the vessels deep inside the tissue, like here 750 microns deep. Remember, the tissue is a highly scattering medium, so this is that's why this technique is actually very significant in the field right now. In the right image, you're actually seeing the neurons, these long dendrites and axons of the neurons. So we're actually imaging using this single neurons as well. And here, basically on the right is a maximum intensity projection. So it's a standard way to process this image. And you can see these big vessels are actually on the surface, and all of these small vessels are going deeper. So this is like a top-down view from the brain, and you're actually seeing like very, very deep levels as well. All of the small micro vessels which are penetrating into the tissue. And all of these yellow dotted are the electrodes, the electrical devices, the electrodes that I showed you. So there are three of them shown over here highlighted. So as I mentioned earlier, that we have these four shanks going inside the brain, so that we can measure from different spatial locations. Okay, and then what I mean by imaging, we can actually do fluorescence imaging, and we can see single neurons firing in the brain. So here is a video basically showing single neurons firing in the brain. So if you notice these very small, tiny spots, they're like flickering. This flickering is actually a single neuron. These are individual neurons that are firing in the brain. And then at the end, it just highlights all of these points where our processing algorithm found these are all the neurons in these spatial locations. So I'll just play it one more time. So this flickering, all of these are single neurons which might be communicating with each other, or processing some information in the brain. Any questions so far? All right. So now
इसमें okay थोड़ा सा if you can तो इसमें जो the sensitivity seems to be pretty good in terms of neuron activity. So can they be used for dynamic to image dynamic problem phenomena as well, dynamics of neurons, और इसकी time limitations क्या है उस हवाले से कि कितनी minimum dynamics ये देख सकता है, क्योंकि 2p 2-photon जो है वो फिर raster scanning है तो it would take time, नहीं?
Yeah, that's actually a very good observation, very good point. So with this technique, we have a time resolution limit. So we can't, we can only measure maybe like 100 millisecond time scale resolutions. So yeah, because of the raster scanning, but to be honest, the raster scanning is actually super fast. The reason why it's 100 milliseconds is because the way we are capturing it, this is not, this is basically a, a fluorescence excitation of a chemical, right? And that
Okay.
has a decay time. That has a decay time of 100, 200 milliseconds. That's why if a single neuron would fire, you would see that the next time it would fire would be with a delay of 200 milliseconds. But if you were to stick an electrode inside that has, and you're sampling at 10,000 hertz, you can measure very fast dynamics using electrical probes. So that's the difference between optical imaging techniques, which only capture slower dynamics, but the invasive electrical recording can capture very fast dynamics to less than a millisecond, right? Because we can have very high sampling rates. So
ठीक है, तो आपकी जो इंटरेस्टिंग टाइम डायनामिक्स है वो मिली सेकंड स्केल में हो रही है, जो जिसमें जिन फिनोमेना में इंटरेस्टेड है लोग न्यूरोप्लास्टिसिटी की प्रॉब्लम के अंदर, या
दोनों में ही, it's in both, because as I said, if there is a brain disease, it would take a day, like couple of, like it takes long to develop. And if we want to study how the neurons are behaving, we really need to electrically record. So that's the good thing about this multimodal acquisition system, that we have all of these modalities simultaneously. So we have all the information, all the data that we need to understand the brain with this setup.
great.
So moving on towards the electrical recording, which is primarily going to focus on these nanoelectric threads and how we actually interpret this data. So here I'm showing an image of what goes on inside the brain. These are neurons, and they can see their different shapes, which basically signifies there are different kinds of neurons. They're just not one kind of neuron, but there could be multiple kinds of neurons. Here I have two probes as well. There is something called an intracellular and another probe called an extracellular. So naturally, our electrode, when it goes inside, there is a very, very, very small chance that it would actually hit a neuron or actually start measuring intracellularly. Intracellular would mean that you're measuring voltages from within the cell. Obviously, it's a very small chance that you would actually hit or go inside the neuron. So most, or 99% of our recording is extracellular, which means we're measuring electrical. All of these voltage traces that I showed were outside the cell. Now, since we have an array, right, an array like a vertical array, therefore we're actually measuring in different depths of the brain. So this is going to be important as well. So here is the raw trace, the local field potential of the low frequency plus the high frequency, and then when you filter out, when you pass it through a high pass filter, you only see these high frequency spikes. And if I were to plot these spikes on a raster diagram, so this is time and this is the raster plot, this each dot would be a neuron firing. So this would be, this neuron fires, so I would put a dot over here. But in this time, you see that this neuron these neurons were not firing, so there is no dot over here. So this is like a raster plot. And if you look at this neuron likes to fire a lot. It has a very high rate of firing, so you're seeing that it's there are dots all around. Now, one of the most interesting problems of these recordings is that we don't have just one of these traces, but we have hundreds of them, because our devices have more than 100 electrodes, or more than 100 recording sites. And naturally, as you can see in this diagram, each electrode would be measuring from multiple different neurons or multiple different cells in space, which could be highly complicated. We don't really know which cell it is recording from, but we can use concepts from machine learning to actually decide or distinguish between different types of cells and different cells. So the way we do this is we actually have this raw data, we filter it, as I just mentioned, with a high pass filter. We then have all the spiking data. And then we do a threshold. You can just threshold this data with an amplitude threshold. So any spike would be above a certain threshold. So you would then detect all of these spiking events. Now, the concept is that each neuron or each different type of cell would actually have a different waveform or a different waveform shape. So over here, you can actually see two waveform shapes, clearly two waveform shapes being overlapped. So this one and the second one goes up and goes down and then goes to baseline. So these, and the reason why you have like so many traces over here is just the average, just trying to capture all of these traces. So this is showing that whenever we see this spike shape, this is one particular neuron firing. And then when we see a different spike shape, we would say a different neuron is firing. So the way we can separate them out is if we do a principal component analysis, or some people also use the wavelet transform over here. So then you can clearly see distinguished clusters in the feature space. So like, what is the feature space? The feature would be, let's say, like, just to naively explain what the feature space would be, it would be the baseline to the peak time, or the amplitude, the time it takes to get to the peak, and the amplitude difference between the peak and the trough, and the width of the spike. So all of these could be different features that you could extract. And once you extract all of these features, you plot them in the feature space, and it could be very high dimensional. And then you would perform simple clustering algorithms like k-means, etc. And from here, you can actually then distinguish two of these different spike shapes. So then you would, whenever this spike occurs in the electrical recording trace, you would say neuron number 1 was firing. And then whenever this spike shape occurs in the recording trace, you would say neuron number 2 was firing. Any questions so far?
अच्छा, इसमें जो लाइन शेप है, so it's mainly because हर न्यूरॉन डिफरेंट तरह से कंस्ट्रक्टेड है, तो जो उससे सिग्नल जो आता है उसका फेज कंस्ट्रक्शन डिफरेंट तरह से होती है.
Yeah, exactly. And not just the neuron type, but because of the geometry, the spatial geometry, because if the neuron is far away, then its amplitude would be smaller. And if the neuron is close to the electrode recording site, its amplitude would be larger. And because of the delay, the shape of the waveform might change. So it's also not just the neuron type, but also because of the spatial geometry in which they are.
क्योंकि reconstruction of the signal would be different अगर spatial geometry ही different है आपकी.
Yes, so this
it's
is
just
how
like a response from, I mean, कोई भी आपका scatterer हो, उसका response different होता है, because I mean it can be different. So scattering problem में भी इस तरह के phase reconstruction different होती है. अगर आपके क्या कहते हैं size, आपके particle ही different shapes या geometry के हों, तो this can be used as one of the indicators for differentiation in scattering problems. Okay.
Right. So the firing activity of, now we can actually, once we have extracted all of these different waveforms, assigned these different waveform shapes to different cells or different clusters, and there could be hundreds of them. So because we have more than 100 channels or recording sites, each electrode on average would record from 2 to 3 different cells. So we would have 200 to 300 cells that we are in different cells that we are recording from. And any two of these neurons or any two of these cells that we're recording from may or may not be independent. And I'll talk a little bit more about this in the next slide as well. So here's just a data processing pipeline that we have. So when we record the raw data, it goes through a pre-processing step, which is explained over here, like the filter that I showed you, high pass filter or a band pass filter, which filters this 250 to 5,000 hertz frequencies. And then there is whitening. And then we have some noise removal and artifact removal, like there are a lot of artifacts or noise in the electrical data as well. They have a very special characteristic. So we remove this, and then we do the spike sorting. So what I just mentioned over here, all of this, it's called spike sorting. So sorting spikes to different neurons or different cells. So this is spike sorting over here, and then we do some post processing that I'll explain over here in the next couple of slides. This is exactly what I was saying. So this is an electrode which is measuring from multiple neurons in space. So multiple neurons would be, when they fire, they would be giving this information to this electrode, this one single electrode. And here again, you're seeing that there are different waveform shapes, amplitudes, widths. Based on these components, we actually found a couple of clusters that I'm showing over here. There obviously there are many more, and here is an image of our electrode. So this is our electrode array. This is one of the shank, and then this is another of the shank. So it's like going deeper. So each block is an electrode trace, and as you can see that whenever this spike occurred, like whenever this neuron, this is a diagram of one single neuron. So the statistics of one single neuron. So when this neuron fired, it always fired in this vicinity. It was always captured by these set of electrodes, but not the top ones and the bottom ones because they're too far away. So here is the average trace of this neuron firing. So whenever this neuron fired, we saw on average this trace, and all of these green traces are the individual spikes. So you can see when you visually, when you see, you can average them and you can see that it looks very similar. So we can also, a very important plot is actually the ISI histogram, which is the interspike interval, and this is going to be extremely important in the next slide as well. So what this means is that as you can see, all of these spiking events, you can compute the difference between any two of the spikes and then plot a histogram out of this. So y-axis has counts and x-axis has the delay between two spikes. This thing, as I mentioned previously in the previous slide, it's a Poisson point process, and you can see it's very, very close to a Poissonian distribution, right? And so what we're seeing over here is that this neuron does not fire with a lot of probability if it has already fired within the first 5 to 10 milliseconds. So this is just the histogram. So how do we actually find correlations between two neurons firing? So we know the firing activity from the spike sorting, and then we can find out the firing activity of that neuron over the entire course of time. We can use multiple different techniques from signal processing, like cross correlation, mutual information, entropy, and phase synchronization. So here I will focus, what we usually focus on, the cross correlation. So a neuron firing is obviously a discrete event. It either fires in each time bin or it does not fire. So the way the cross correlation is computed is very similar to a convolution. So here we can see that there is a reference spike, and then you would create these different time bins. And if so this target is one cell and reference is another cell, and you're trying to find the correlation between these two cells. So if this, if you look at this one spiking event on the reference cell, then you would create these time bins, and then you would add to the histogram, add counts to the histogram whenever the target cell fires. And then you would move to the next spike in the reference cell and do the same thing, and you would get this histogram. And you would do this for all the spikes in the reference, and then you would create something called a cross correlogram, something, it would looks something like this. So on the y-axis, you would have counts. On the x-axis, you have the time delay in milliseconds between the spikes. So naturally or intuitively, we know that if we can understand that if two neurons, their neural activity is not correlated, then it would be flat. The cross correlogram would be flat because the probability of firing, if one neuron fires, the other neuron does not have a preferential firing time to fire. But if two of the neurons are linked together, or if they have strong correlations between them, let's say they have a direct connection between the axon and the dendrite between them, then in the cross correlogram, you would see a very strong peak at some certain delay. And now there are different definitions. I will just show here on. So here what I'm showing is a cross correlogram between two neurons. So here you can see all the counts, the blue, right? And this red is a certain standard deviation threshold. And you can see that these red bars over here, these long red bars, in the cross correlogram, this thing crosses the threshold, which is an arbitrarily set threshold, and it crosses the threshold at the delay of around 2, 3, 4 millisecond time bins. So if this happens, neuroscientists tell us that this means that there is a direct connection between the two neurons. So they are like directly connected. We call it monosynaptic connectivity. And another concept is synchrony, which would mean if this thing does not happen, we can also compute like how synchronized the firing pattern of two neurons are, or just counting the counts in these time bins. So with this definition of connectivity, or like defining a connection between neurons, and remember after spike sorting, we could find hundreds of neurons or hundreds of cells or hundreds of clusters. So then we can actually form a graph of connections, or a connectome of all of these individual circles are the neurons or the cells that we found. And then a line would represent that there is a monosynaptic connection between them. And then we can also find the direction of the connection, because if this large peak occurs in the positive delay, that means it's a connection in one direction. And if this peak occurs on the opposite side with the negative delay, at the negative delay, then that means that the connection is in the other direction. So this is why you can see arrows as well over here. So this is from our data where we actually found the connections. So this is the same diagram, but more geometrically precise. So these different vertical columns are the 4 different shanks that we have of the neural threads, right? And all of these circles are the neurons that we found, and we can see that there is a lot of connections within the same column, and very few connections across different columns. Any questions so far?
Not yet. Can you finish maybe in 4 to 5 minutes? Haad?
Yeah.
Okay. Yeah. So this. So one question. You do have you do have Poissonian statistics for single neuron. Is that correct?
Yes.
But then you have correlation between different neurons in between. So what does a individual Poisson statistics would mean? I mean, in terms of firing out, like, okay, if it's shown, what does it mean that agar ek neuron fire like karta hai, like there is no kind of क्या कहते हैं preference and like कोई वो नहीं है, क्या कहते हैं उसके अंदर कोई statistics उसकी completely randomized है. That's what it means, na?
Yes. Yeah, I can answer. I understand. So it's a very valid question. Basically, Poissonian is so there are 2 problems over here. The first one is that the neuron is not truly Poissonian, right?
Hmm.
Poisson is a model that we use, but it's not truly exactly Poissonian because of the refractory period, right? The second thing is that it's only well modeled as a Poissonian if it's spontaneously firing. A single neuron is spontaneously firing. But as you said, if there are two neurons that are connected, they wouldn't be Poissonian, right? Because there's interdependence, and then they are
Yes.
not like memoryless, you can say, or they're not independent.
Yes. So वहाँ पे फिर Poisson work नहीं करेगा. Or it would disturb the Poissonian statistics in one way or another.
It would completely destroy the Poisson, but the Poisson model is like a good model to use for a single neuron spontaneously firing. Like you can also imagine that if I were to plot the rate of firing of a neuron with respect to a stimulation, then it would not be Poissonian because it would be
it would be highly
with
okay. Yes, sync with whatever the stimulus is then. Yes, or the statistics of the stimulus. Yeah.
So I only have a couple of minutes to talk about my research work. All of what I talked about was background or techniques and code development, data analysis that we do. Just a couple of slides on brain plasticity, and this is what I'm working on. So here I would just show what we have over here is a mouse which has these whiskers. And the way these whiskers are actually organized is that each of the whisker on the snout of the mouse is spatially mapped to a different region, localized region in the brain. And if I zoom in, you can see each of these whiskers they're labeled in the literature as like these labels A4, A3, blah, 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 right? And then you see that in the brain, they have very localized spatially localized. So if I were to actually stimulate one of these whiskers, you would see that there is a very sharp and short-lived neural response, which is the electrical response using our electrodes, neural threads. And then there is a slow increasing blood flow response. So what happens is that when you stimulate the brain, like let's say this whisker and then this barrel, the electrical activity is very fast and short-lived, but the blood flow activity is very slow. I will just show, this is on the top, you see the electrical activity. You can see this is the change in the power spectral density at different frequency bands. So you can see that the red line, the first red dotted line is where the stimulation began, and almost instantly we see the activity come up. But here is the speckle imaging that is being shown after the whisker stimulation. And you can see it's slow and it's long-lived, and then it only occurred in a very localized region, exactly like I'm showing over here. So when I'm stimulating one of the whiskers or one part of the brain, only that part of the brain, the blood flow or the hemoglobin concentrations would rise in that part. As I mentioned in the previous couple of slides about spike sorting, we can also extract single neurons firing. So here I'm plotting as a function of time the firing rate. So the number of spiking events divided by a delta t time. So here we're seeing that almost instantly, as soon as the stimulation began and the red dotted line, you see the firing rate of 1. This is one single neuron jumped up, and then slowly it remained high up until the stimulation was on, and when the stimulation turned off, it basically dropped back down. So what we basically do in this project is that we study brain strokes. So brain stroke is basically where you would destroy a certain region of the brain, and then you would try to understand how the electrical activity and how the concentrations of hemoglobin and how the blood flow is changing and how it's actually recovering. So there are two kinds of strokes. The first one is the ischemic stroke, which is where blood vessels are blocked. And the other kind of brain stroke is a hemorrhagic stroke, where a blood vessel is burst. So 85% of the strokes are in human beings in the brain are actually ischemic. So we are studying ischemic strokes. So ischemic stroke is the blockage or reduction in the blood flow. And these are our electrodes inside the brain. And then this is just a screenshot of, a stroke is actually on one of the cortical columns. So let me just finish off over here by showing you some of the results of how we do a brain stroke and what happens after a brain stroke. So what happens after a brain stroke is that the brain naturally would try to recover and try to fix the effect of the stroke. So the way we stroke is again using an optical system. We have a high-powered 532 nanometer laser, which induces a photochemical reaction in the vessel. And the way we target this is using a DMD. So a DMD, we can actually project a beam onto a very specific shapes. Like here, I'm actually targeting these green regions, right, in the brain. And we are basically blocking flow over here. And the way we do this is by basically we know the image, we can see the image, we can draw a pattern over here, and then we can find a transformation on the DMD, like what pattern do I have to display on the digital micromirror device so that the green laser shines exactly on this pattern. So this is just to show this is the pattern of a bull. And we are seeing over here that the green laser would block flow in all of this region and not block flow in the other regions. And I have a short video of how using the green laser we are actually blocking the flow. So this is a vessel, as you can see in the video on the bottom right. This is a vessel and it will quickly go away. Yeah, you can see over here it went away. So this is basically what we're doing is we're blocking the flow over here using this high-powered beam. So you see the vessel actually goes away, and then this is like a very high, fast forwarded. So we can see the vessel goes away, and then the blood flow is blocked and the neurons would die over here. We have successfully induced a stroke in the brain. And then our study begins where we start to see the effect of the stroke. So we have all these tool sets of electrical recording, spike sorting, and then the imaging. So we record all of these different modalities and see what goes on inside the brain. Just to give you a quick, I think I will basically end with this because maybe you guys are running out of time. So this is the plot of what goes inside the brain after a stroke. So here I have two plots showing the LFE, which is the low frequency electrical activity from our neural thread probes, and the red plot is the CBF, which is the blood flow from the speckle imaging. So you can see that they are very strongly correlated in a normal brain, which is baseline. We have not introduced any stroke in the brain at this point. And the red dotted line shows where the stroke was introduced. So as soon as the stroke was introduced, you can see that the LFP, or the neural activity, the electrical activity inside the brain, quickly dropped. And we're tracking this longitudinally because that's representative of the title of the talk, was that we are actually recording from the brain longitudinally, for a couple of weeks and months. So you see the electrical activity actually died down. The blood flow also died down on the day of the stroke, but it quickly recovered in the subacute phase. After a long period of healing and recovery, this is plasticity, where the brain would actually start to form new neurons, new connections, and then start to grow back all of the lost function. And then we can see that the blood flow and the neural activity actually become correlated again, which is the recovery stage. But the most important thing is that actually during the subacute phase, where the effect is the strongest, they're completely anti-correlated. And this study and this finding was extremely, this was done in 2020 Science Advances paper. And this is extremely important because all of the brain imaging, like human brain imaging that we do when we go to have an fMRI, they're not sticking electrodes inside the brain, and they're not measuring neural activity inside the brain. What they're measuring is the blood flow activity. So fMRI is measuring the changes in the hemoglobin concentration or blood flow. It's proportional to the CBF. So this was extremely important because we saw for the first time that they are completely anti-correlated. So you cannot trust an fMRI exam during the subacute phase. In a normal healthy brain, yes, the fMRI would give you very good results. In a recovered brain, they would also give you very good results. But during the diseased part, you cannot infer neural activity, the LFP, from the fMRI or blood flow response. That's my conclusion. And I think I have a couple of more slides, but maybe I can upload the slide deck for you guys to see.
Yeah, okay, great. Yeah, thank you. It's such a nice talk. Okay, great. So yeah, very interesting results. Yeah, so you have managed to separate the electrical signal from kind of the hemoglobin signal, and that give you really an insight into the neuroplasticity problem of the brain. That's so the question is, how would you address, for example, the evolution of neuroplasticity with aging, for example, or even longer time scale? I mean, you hinted it at the very beginning of the talk. Us tarah ka problem, do you have that in mind? And if yes, how are you going to approach that in this kind of yeah?
So there are many. I think aging is also something that we can do with our devices and with our imaging systems. The advantage is that these flexible electrodes can, they last inside the brain for a very long time. Most of the other research groups are using rigid electrodes like silicon probes. So they cause a lot of damage to the brain, and then they don't last for more than a month or more than a couple of weeks. So these electrodes can last for more than a year. So you can actually see the neurodegeneration of the aging effect. And one of the most interesting studies for aging is actually Alzheimer's, because Alzheimer's is memory loss. Like over time, slowly there is memory loss as we age, and that's something people are studying, but our lab is not doing Alzheimer's models, but the people in other research areas are actually studying Alzheimer's.
Okay, great. Question. So oh, okay, it's a very nice talk. So if someone wants to go, let's say, from physics, I mean, अगर कोई undergrad है अभी या वैसे graduate student है physics में and wants to move into this direction, इस तरह की, let's say, like क्या कहते हैं, इस तरह के problem की तरफ, let's say, जाना चाह रहे हैं. So what kind of skills would be, you think, are important to develop as a physics student?
Yeah, I think I was also a physics student at one time, and my entry into this field was using my optics skills. So I had a lot of experience with both theory and experimental optical imaging skills. So that's how I actually entered the field, and I started developing these imaging systems that I showed or described above. And then actually recently I started working on the electrical activity as well, and applying the signal processing technique. So this is how I naturally entered the field, I think.
I mean, these like techniques are quite data intensive as well. So you might also, it's good to have data.
Yeah, very, very data intensive.
Skills. Yeah, highly data intensive. And apart from statistical analysis, correlations, data, other kind of data analysis technique would really come in handy if someone wants
Yeah,
to
I
go.
think having a good understanding of statistical theory would be very useful, like stochastic processes. I think there is courses at LUMS on this, and
Yeah.
the data, I think like obviously MATLAB and a lot of Python these days, very common for all of this analysis and code writing that we do. And as you said, it's very data driven. As a matter of fact, each like 1 30-minute session of recording is around 150 gigabytes of data. So only 30-minute
Wow.
recording when I do using, because I'm capturing at 70 FPS, like four different imaging modalities or cameras, and then you have 30 kilohertz sampled electrical recording from 100 channels. So you can
Wow.
imagine how
Okay.
much data. And actually, as a matter of fact, we use 3 different PCs because the data rate is limited. So we need to,
Yeah.
when we're doing the experiment, we need to like separate out the data streams because the USB does not have that much capacity.
Okay. Yeah, there's quite a lot of quite intensive in that sense. Okay. Yeah. So any question, Josefa? You have question or so? Yeah. Okay, if not, then yeah, but thank you again, Haad, for sharing such nice work and kind of opening up some new directions which one like a physics student can go into. I mean, these are really interesting problems, okay, which are being applied. अब जैसे जितने सारे optical techniques आपने बताया, on top of that you have these complementary electrical techniques, and they really add up to give you a deep insight into such a complex problem. And that's where the benefit of working in a physics degree comes from, that the kind of training you get in a physics degree can really
Yeah, I
help a lot.
definitely agree.
Yeah.
As a matter of fact, both of my PIs that you can see, I have named them as well, and in the picture as well, they're all actually physics PhDs from Stanford. And as a matter of fact, almost like 30% of the professors working in this field are actually physics PhDs. So, yeah, I don't know why. Some of them are electrical engineering, but very like you wouldn't you would like, it's not like they're all PhDs in neuroscience or neuroengineering, but most of them are actually outside the field.
Yeah, great. Yeah, okay. Yeah, with that note, so yeah, thank you again, Haad, and yeah, so hope we can invite you again sometime in future. And if you are visiting here, you definitely should visit. Yeah, the LUMS again, and we can have a chat. So yeah, thank you, Haad. So,
Thank you, Dr. Azwan, and thank you everyone for joining in the talk.
yeah, yeah. Okay, okay. Assalamu alaikum.